Okay. So what I wanted to talk to you about is, first of all, how AI can advance digital pathology, but also the context in which digital pathology sits today. So we can basically look at H&E. These are more or less the slides you are familiar with standard publications. And here we can improve diagnostic reporting. And of course, what a lot of you have also looked at is IHC, that's basically immunohistochemistry of specific proteins, and that is usually used to make an analysis more robust and more reliable, and I give you an example of this. And then what I also want to mention is there are new imaging modalities that basically allow us to look at multiple different proteins in the same tissue section. And then, of course, we want to combine this with RNA and DNA expression, and I give you an example of this during this talk. So what I want to do is basically go a little bit through various different applications or give you an idea or give my almost like perspective on how AI can improve the field. And in terms of morphology, what's really happening at the moment, especially in the UK, we have sort of national initiatives to advance digitization of pathology. So we are part of one of those. You can call them, they're a little bit like NIH roadmap centers. They're here sort of Innovate UK funded centers where this enabled, for example, Oxford to become fully digital just about around a year ago. And there's also now a focus on training pathologists and using of digital tools, and of course, on the computational side, train doctoral and postdoctoral AI researchers with respect to these applications. There's a little bit of the question, what are the near-term benefits? What are the short-term benefits or the longer-term benefits? So one example I want to highlight today is basically the reduction of waiting times for results and also workflow improvements. This is something we think can be implemented very soon. And maybe we can also discuss at the end on how reliable AI solutions are in this kind of setting. I'm sorry this is not the greatest figure here, but this is a copy of Robert Virchow's cellular pathology book. And he's basically stressing the fact that the cell is really the ultimate morphological element we should look at. It manifests changes of life. And he was sort of a founder, or is considered the founder of modern pathology. And what he did at the time, he started drawing these pictures of cells. And of course, he started to almost like illustrate how cells change in the context of disease. And that is actually sort of leads to my first example. It addresses the diagnosis of myeloproliferative neoplasms, which are forms of blood cancer. And this is work here led by a pathologist called Daniel Royston. And in that context, megakaryocytes, which you see on the right, these are basically larger cells, platelet-producing cells, play a significant role in diagnosing the disease. And to give you an idea on what's been described in the WHO guidelines that pathologists basically are being trained on, there are basically 6 different classes. These classes are different with respect to size and basically appearance. And the names that are sort of given are loose clustering, dense clustering, cloud-like nuclei, and Staghorn nuclei. So the point I wanted to make is that language is actually a very limiting factor in basically teaching and also reporting pathology, because this particular WHO guideline is basically has a pure level of reproducibility, which has sort of been shown in different studies. So the workflow we developed is sort of, more or less imply the tools of detection and segmentation most of you are familiar with, to build a large annotated library of megakaryocytes. So we set ourselves the goal initially to start out with the library of 20 to 30,000 megakaryocytes. So we start off with basically an unlabeled pool of biopsies. Then we use, deep learning-based detectors like YOLO detectors to detect megakaryocytes. And here we are quite careful in the sense that at every step, we essentially get feedback from pathologists that that more or less ensures that at least two experts agree that this is a real megakaryocytes. Then we do the same to the segmentation. Again, there's a validation loop in here where pathologists confirm that this is a well-segmented megakaryocyte. And then we built a library of these megakaryocytes using clustering methods. And using these identified clusters, we now go back and we sort of annotate the slides and we look at different disease states. So the point I wanted to make is that, first of all, the annotation of these slides, if you really want to gain high throughput, needs a little bit of thought. So just drawing around the cells is very slow. This is what you see at the right. And we also looked at input with styluses and stuff like this. But if you actually have a classifier that performs reasonably well, then you can essentially highlight the megakaryocytes, and then you just ask experts for confirmation. And that really allows to basically scale up a library and also basically use the time of experts wisely and efficiently, and come back to that point at the end. So here, in terms of the feature extraction, we're really doing nothing extraordinary. We're basically using an autoencoder to basically capture the features of megakaryocytes. Then in order to make this more robust, we basically also do vertical flips and horizontal and vertical flips to make the feature vector somewhat more invariant. But what you see here, although we train on reactive and a case which is called ET, the reconstruction error for the other two disease subtypes, PV and MF, are actually within good expectations, so we can basically use that representation. What we can now do is we can essentially use these 9 identified clusters, and now we have a data-driven basically approach to these phenotypes, where you basically saw before we had 6 phenotypes that were sort of loosely defined. What we can now do is we can take essentially a slide and we can characterize each of the megakaryocytes as basically belonging to one out of those 9 phenotypes. And then we can also look at this, we can look at various other features like the propensity from other clusters, the density of cells, the average cell radii, and can basically build up a fairly quantitative and interpretable descriptor. Now the question is, what do these actually representations help for the understanding of disease? And what we did here is, initially we thought the histograms are actually already very helpful. What turns out is, if you overinterpret the histograms over the phenotypes, it is often misleading in terms of when you want to relate two cases, or when you want to basically look at a disease progression. If you, however, look at basically the distance between two histograms, or the evolution of two histograms over time, there's some very valuable information here. So here, what you see on the right are basically first and second order principal components, where we basically project changes in that phenotype signature. And what you see at the top, you see two patients with stable disease. And what is really nice here, we can actually map the entire disease space, and you see here that, for instance, the blue icons basically stay in roughly the same area, as well as the red icons, which also stay in the same area. And these are samples that are roughly taken over basically 2 or 5 years. What you see at the bottom is a very different case. These are two patients where disease actually progresses. And you can see clearly there is a shift in this basically disease map, from which is quite clear that the patients basically move from sort of the PV state to the green, the MF state, and that's actually more aggressive disease. So this representation is now, of course, terribly useful to look at entire clinical trials, or to also look at basically predicted response to therapy. So this shows that capturing morphological information in a more exact and robust way is actually extremely helpful in analyzing entire patient cohorts. So the next case I wanted to highlight is, remember what I was saying in the beginning, what can we really do for clinical pathology in the short term? And can we, for instance, look at the connections between morphology and protein expression? And here it is sort of quite clear that IHC labeling is already used to diagnose ambiguous cases. So today, basically, a pathologist would order IHC if he is unsure. But one problem is that this disrupts the workflow, in the sense we study this in the context of prostate cancer. So imagine somebody looks at a case and he says, well, now I need IHC. That basically the case goes back to the lab, and a couple of days later, the entire basically slides come back. And the question we asked here is, can we actually learn, can we predict when IHC is required? And here are some examples that just illustrate to you where pathologists use basically additional protein stains. So what you see on the panel A is here, well, there are some short glands. They're likely to be cancer, but they're really quite short. So they're basically, their spatial extent is quite minimal. When you see in panel B, if this is now stained with CK5, these are sort of clearly confirmed as cancerous glands. And what you see in panel C is a small focus of suspicious glands. And again, in panel D, you see that these are confirmed with the help of IHC. So this, you can look up the paper. This just was published very recently in Modern Pathology. And what we basically did, and this is almost like more, the equal amount of work went into annotation and modeling than almost like the algorithm development. So what our pathologists basically did, they carefully went through the reasons why they order IHC. So you see here, short length of cancer, that's case what I just showed you in panel A. Unusual morphology that are difficult to classify is another example. So I'm not going to go through these very specifically, but I just want to sort of illustrate to you what upfront work we did jointly with pathologists before we actually did annotation and then subsequently learning a model. And of course, the goal here is to predict diagnostic uncertainty. So it's either clearly benign, it is ambiguous, or it is clearly malignant. And we are really interested in sort of this ambiguous category. So the model we basically use is, we use a tile-based uncertainty predictor. So we split the biopsy into different tiles. And then we train a residual attention network to do sort of this tile-based prediction. So basically for every tile, you get a confidence score, whether it's ambiguous or certain. And then we combine these to a whole slide IHC request predictor like you see it below. So we basically then use a tree-based classifier to predict whether IHC should be ordered or not. Just to give you a better idea how much data was used for training and validation. So we basically used data from 6 months, and we worked closely with 3 different pathologists. So we had 241 patients, 23 control, 218 IHC requests, where each slide was annotated by only 1 pathologist, and that was basically used for training and testing using basically these 3 splits. And then for validation, we had 222 needle core biopsies that were selected from the recent hospital archive, and we did the prediction, and then we compared to 3 pathologists, where basically each pathologist annotated all the slides. So when you look at it, what are essentially sort of the potential improvements? The mean turnaround times for IHC requests, at least in the NHS hospital we are affiliated with, was 7 days and 10 hours. The mean turnaround time for the non-IHC request case was 4 days and 5 hours. So that's basically, in terms of reporting, a potential saving of 3 days and 5 hours. And of course, then we also looked at the estimated time per pathologist, and that turned out to be around 12 minutes per case. Now, we don't argue that this is a universal model that can be applied without optimization at different clinical sites, because local guidelines might differ. But we just wanted to make the case that a tool like this can actually lead to workflow improvement. And what I also think it's a very nice case to now study: how do actually AI tools behave over time? How robustly are they in the clinical workflow? So that's one of the next steps. We want to use one of those algorithms to actually use them in the clinical workflow on a day-to-day basis. And in terms of the analysis of the learned model, the tile level accuracy ranges from 86, 85, and 81% based on the different splits of the data. Here you see the visualization of what areas of the tissue influence the decision whether the slide is ambiguous or non-ambiguous. And remember, in the validation, we actually compared to 3 different pathologists. So you see 3 different ROC curves, and you also see the test, train, and validation sets visualized down here in panels B and C. This gives you an idea what the modified workflow looks like. So the biopsy arrives in the lab. The slide is scanned, and the scanned slide is now basically used, is analyzed directly, and the basically IHC is requested, and the pathologist only sees the IHS when the case when both the H&E and the IHC are available, and that's basically leads to the time saving I illustrated before. So the next case goes a little bit further, and we're trying to sort of now investigate whether we can link morphology with basically molecular information, especially here RNA expression. And this is something here we really want to sort of investigate. Can we do something that pathologists really cannot do today? So what are more or less the opportunities for next generation pathology? So what we're looking at is, in a case of colorectal cancer, we look at the common molecular subtypes, CMS 1, 2, 3, and 4. I'm not going to go into the detail of their biological relevance, but it suffices to say that basically CMS 4 indicates worse relapse-free and overall survival. So these are basically cases we would like to identify, and the CMS classification, which is done purely on RNA sequencing today, is also part of the WHO recommendations now. Here we have 3 cohorts we are analyzing. So we're using basically one for training, which is called FOCUS, around 360 patients. And then we have, of course, a TCGA, which is publicly available to everybody else. That's 375 patients. And then we also have a local cohort of basic rectal biopsies. And this is quite significant because these are biopsies where there's a lot less tissue available. And we have 183 patients. So you see here, these are 2 millimeter biopsies, whether the other are resection specimens. We also have ground truth based on RNA sequencing, and you see here that in all cases we have some unclassified samples. These are basically samples where the RNA expression is not sufficient to call one of the classes. And you also see that in the Grampian case, remember these were the endoscopic biopsies, we have far more unclassified samples. So initially we thought, well, one of the pathologists was using HALO, a tool some of you might be familiar with. We do feature extraction and classification and everything will be easy. But of course, this didn't work at all because we have actually very small changes in the tissue, which are almost like swamped in a sea of other features. And I'm basically coming back to that point at the very end of my talk. But here we sort of applied a domain adversarial training technique to make sure that we don't learn features that are specific to one given cohort. So we use the labeled samples from one cohort. From the other 2 cohorts, we essentially just use unlabeled tiles to make sure that we don't learn any features that are specific to one cohort. We learned 5 different models. These are now ResNet 50 models. And at the time of inference, we basically take a patch, run a patch-based classification, regenerate this overlay, and now you already see that we now have a localized overlay where pathologists can actually see if there's a mixture of classes present. So here you just see our training data. We basically here the tumor area is annotated by pathologists by hand. This has now been subsequently automated, but here the entire tumor area is used for sequencing, so we don't really know which of the tiles belong to what class. We only have basically one class label. After domain adversarial training, these are sort of the performance scores we get in terms of concordance with sequencing that range sort of from 82 to 89%. We get lower performance on TCGA, and this has good reasons because the sectioning quality between H&E sections and sections that are used for sequencing is not as good as in the cohorts we have locally. Here you see a result where you see the morphological association between CMS1, and you actually see that in this case, we see some presence of CMS4. So this means that now our pathologist can actually look at some of the basic heterogeneity in the tissue. This is an example which is a lot more homogeneous. And here's another one, which is also CMS3, which is more homogeneous. So of course, you could basically use the traditional sort of gradient-based visualizations. And here we did this just to basically get an idea. And CMS1 is basically known for features in mucin, lymphocytic infiltration. That seems to be picked up by the model, but we sort of don't interpret these. We just sort of recognize that this is something that's sort of plausible. And again, in CMS3, we see mucin-filled glandular structures, similar sort of result, which we're sort of quite satisfied with. What is far more interesting is then we now look at the different cohorts and we see which patterns are present. So we, for instance, basically just look at all the morphological clusters. We split them more or less randomly into 13 clusters. But you will see is that in each of the three different cohorts, you see similar histograms for CMS1 and for the other CMS classes. And that really means that the sort of morphological information we can extract is actually reasonably stable. And the point I wanted to make here is that we can take samples that were previously unclassified. Remember that on the RNA expression, we can't classify some of the samples. And we can go back and we reinterpret the sequencing results. And here, for instance, what you see in gray, we can take a CMS4 case and look at its KRAS mutation. So this is quite significant because now we can essentially use morphology and the sequencing information in tandem. So at the end, I wanted to make a couple of comments which I sort of label under the heading doing more with less. And this relates to the fact that we really cannot do always have complete annotation of the data. So here we basically, this is work that was published in MICI last year. We look at fine-grained distance classification through deep attention networks, where of course this has been explored in the vision community already. But here what we said is, look, in medical imaging, we often see many instances of something. And we wanted to sort of extend that and optimize it for biomedical data. We also wanted to use the attention maps to see if images can become more interpretable when these attention maps actually highlight what features it's being used. This is sort of the challenges that are being addressed here, the limited size of data sets, really expensive annotations because we really use precious time from clinical researchers, and we have a lot of high resolution information, and we want to use it multiple discriminative instances. So here's basically, this is the network. We have an input image, we have generate a low resolution image. We basically have sort of the typical CNN pipeline for downsampling. We have a high level semantic feature space. We have the map. And then what's really the innovation here is this using a gated mechanism to generate that attention map. And then we also basically almost like upsample the features that are important to make a decision. So here we really don't use any pixel level annotations. These are all basically slide-based or region-based annotations. And this is just for reference. This is how it was compared on the fungi data set with other networks. And at least at the time, this was a very attractive and also well-performing method. And you see here that we can also link the attention maps with the semantic interpretation of the specimens. And the interesting thing is then we use this on the Bach data set, which is a publicly available breast cancer data set. And we wanted to check if the highlighted areas actually coincide with areas a human pathologist would look at. So Meng Guanfan, who did all this work, basically took these regions and she compared them with annotation from a pathologist. She basically gave the same slides to a pathologist and say, if there's one region in the image, which region is basically the most important one for diagnosis? And between the basically highlighted maps and the rectangles you see on the top, there was a 75% concordance that basically for that limited study, we thought was very encouraging. And then finally, I just wanted to give you an illustration on how we now apply this in other conditions. So here we're looking at celiac disease. And one problem in celiac disease is actually to find villi. So imagine you have the basically epithelium, sort of the inner skin lining of the gut, which has these little villi. And of course, if you cut a specimen, you cut these villi in different angles. So you now want to find which villi can be used for interpretation and diagnosis. So here, very similar principle. We use a tile-based approach. We use essentially this attention map. Here we only have case level labels. Here you see the results. So the feature maps that were generated by ResNet are shown in the middle, and they're shown entire regions. And then you see our attention maps shown on the right, and they're actually far more specific in terms of villi locations. You see the performance of the of the model below, and of course in the intermediate classes, for instance, the March type 3C, it's not performing incredibly convincingly. So this is something we're looking at the moment, but there's also the tendency that pathologists actually use the mid-grades far less. So there is a class imbalance problem here as well. And you see the confusion matrices. So there is basically more confusions between the sort of Marsh types 3 than basically the Marsh types 1 and 2 overall. Some final thoughts. So I think subtle changes are, of course, something pathologists are highly trained in. They are in small areas, and they basically need to be detected as these can drive disease progression. Given the size and complexity of these images, it's very difficult to do basically exhaustive pixel-based annotations. And what I've shown you with the CMS4 case is that, or CMS case, is that computer vision algorithms are certainly now in the position that they outperform humans in terms of identifying differences between sort of fine-grained patterns that really humans cannot tell apart. But there's one important caveat, and I think that's an important area of research still, is that pathologists are actually trained where to look on the slides and what area to basically, what information to ignore. And that's often where automated algorithms fall short, because they essentially combine irrelevant information with information from really diagnostically important areas. And this concludes my presentation. I'm going to open the floor for questions.
Thank you again. So we have time for 2 questions. So somebody can use chat, or maybe can just directly ask Jens.
Hi, Professor Rittscher. So thanks for the great talk. I think one of the questions that I have is that, other than the features of the cells, there is now increasing attentions on the investigations of the roles of other tissue components, such as the stroma and the collagen fibers in the prognosis of the disease. So do you think a more comprehensive model that models the relation and the interaction between the cells and the microenvironment would help better explain the observations or help with the discovery of, let's say, new patterns for diagnosis, or even provide guidance for like a patient treatment? Thanks.
Yeah, I think you're making an excellent point. So I've selected examples here, but of course now setting basically almost like different tissue architectures in context, yeah, and use the hierarchical arrangement of the tissue. And I think you mentioned the stroma. These are really, really important questions, and especially looking at the microenvironment, right? Because I think using these types of algorithms, we provide sort of a new way of combining different features, yeah? So what we are, for instance, doing at the moment, CMS is a global model. We now basically refine this to look at tissue-specific signatures. So for instance, we say, what does actually CMS features look like in the tumor, in the stroma, and on the front? So I think you're absolutely right. This is sort of almost like the next level of analysis, where we can basically look at these different components and also then look at different sort of almost like protein expressions.
Oh, thank you. Thank you so much.
Yeah. Anybody else have any other question?
Have you seen that there's a large sensitivity to the downsampling resolution that you have? So you talk about, in the case of the fungi anyway, and I assume also these slides, that you're doing some downsampling as your input. Have you looked at sensitivity of downsample to identification of microstructure, basically?
Yes. So in each of the experiments, we typically do an analysis on how the image resolution affects the results. So for example, you see that many slides are diagnostically scanned at 20x, where often pathologists insist that certain cases should be looked at basically 40x, and you see there is a change in performance in some of these algorithms, especially if you look at cellular features. Yeah. And if, for instance, in the case of CMS, we have documented this, and you see that in the supplementary material of the paper. But it's quite important to basically see how image resolution affects the result, because I think it's sort of intuitive, because image resolution and the complexity of the tissue architecture are basically always have to be kept in balance, right? If you look at features that are not as fine-grained, well, then you can downsample. But if, for instance, if you start looking at intracellular features with some, in some diseases you do, then of course any type of downsampling will degrade performance.
And then I guess my next question becomes, if right now this is what I guess I would say is manually done, right? Like you are choosing multiple resolutions, and then you look at them all and your model performance. Have you thought about integrating like an automated scaling transformer as like an input layer or something like that to your network?
Well, we've basically looked at a few years ago at basically models that integrate the scales. We basically tested various models at the time, but I think what your suggestion is definitely something that's interesting to follow up on. We haven't done it in that systematic way of basically doing this basically a scale transformation as you suggest.
Cool. Okay, thanks. Yeah, this was great work. Sorry, I should have led with that, but yeah, it's really great work.
Okay, with that, I would like to conclude the session, and thanks, Jens, for the very nice talk. What you presented is what's happening right now in blood tests, all robotically done in most universities, so that there will be some day where all the histopathology tests will be done mostly robotically, not to replace the pathologists, but at least to give them all the tools they need and potentially do this automatically. So all these methods are keep evolving, and I think there's a great future in the use of AI for histopathology. Well, thank you very much. So now we'll move to the regular paper presentations, where every paper is a total time of 15 minutes, but your talk should be 10 to 12 minutes max. You'll have questions and also follow the schedule. So I'll warn you if you go over. So the first paper is titled Unsupervised Detection of Cancerous Regions in Histology Imagery using image-to-image translation. And Dejan Stepek from XLab is going to give the presentation.
Thank you. I hope that you can hear me well.
Yeah.
Okay, thanks. So hi everyone, my name is Dejan, and today I'll present my research work on unsupervised detection of cancerous region in histology imagery. I think